Hello everyone, I am going to explain about applications of biotechnology in industry. Applications: improvement in fermentation products, microbial production of synthetic fuels, microbial mining or bioleaching, microbial biomass and single cell protein production, production of enzymes and human proteins, production of secondary metabolites from cultured plant cell. First one, improvement in fermentation products. This achievement can be done in different ways by selection of improved strain, by transgene application into the microorganism, by using cheaper raw material, by manipulation of medium constituent, as well as by simulation of the reactor and the adjustment of different cultural conditions like pH, temperature, etc. Products of microbial fermentation include primary metabolites, secondary metabolites, enzymes, proteins, capsular polysaccharides, and cellular biomass. Next, microbial production of synthetic fuels. Important fuels can be produced by using many microbes, which include ethanol, methane, hydrogen, and hydrocarbons. Zymomonas mobilis produce ethanol twice as rapidly as yeast from carbohydrates. Methane, which is used in various industrial purposes, can be produced by Clostridia, bacterioids, Scleromonas, etc. from the waste. Next, microbial mining of bioleaching. The process of bioleaching requires metals from one ores which are not suitable for direct smelting because of their low content. The application of bioleaching process is of particular interest in case of uranium ore. Thiobacillus peroxidans is the commonest organism which involved in case of copper and uranium-1 ore processing. Microbial biomass and single cell protein production. Microbial production of commercial significance is the microbial biomass. Example: commercially produced yeast cells, bacteria flavoring cheese from fungal biomass. Single cell proteins are the derived cells of microorganisms such as algae, certain bacteria, yeast, mold, and some higher fungi. The protein percentages for every single cell protein are high. Production of enzymes and human proteins. Bulk of the enzymes is obtained from microbial source of fermentation process. Now several plant enzymes are being used in papain from Carica papaya, bromelain from Ananas communis, ficin from Ficus glabrata. These enzymes are used in meat tenderizing as protein hydrolysates in beer industry in clinical application. Recombinant DNA technology can be used for the improved production of enzymes and proteins. There are many human proteins which have long been believed or known to have therapeutic potential, and their increased production has been achieved by using recombinant DNA technology. The gene coding for insulin with two peptides or polypeptides has been synthesized. Each synthetic gene was linked to a plasmid near the end of the beta-galactosidase gene of E. coli. After the gene expression and the translation of mRNA into protein, the two polypeptides were cleaved from the enzyme and linked to form the complete insulin molecule. Different workers also synthesized complementary DNA from RNA of rat pancreas with the help of reverse transcriptase, which was inserted into PBR, the 322 plasmid in the middle of the gene for penicillinase. The plasmid also contained the structural genes of proinsulin T. The hybrid protein synthesis in the bacterial cell was penicillinase plus proinsulin, from which insulin could be separated by trypsin. Production of interferon took a vital position when human leukocyte interferon was engineered by yeast cells. A DNA sequence coding for human leukocyte interferon was attached to the yeast alcohol dehydrogenase gene in a plasmid and introduced into cells of Saccharomyces cerevisiae. The first human peptide hormone synthesized in a bacterial cell was somatostatin, which is one of the group of hormones secreted by hypothalamus, controls the release of several hormones from the pituitary. The synthetic gene has been inserted into the plasmid. Expression vector was constructed from the plasmid PBR322, to which was added the control gene region and most of the beta galactosidase gene from the bacterial lac operon, and the gene was inserted next to beta galactosidase. After the plasmid was introduced into the cells of the bacterium E. coli, the hormone was synthesized as a short peptide tail at the end of the enzyme. This is the diagram for the production of recombinant insulin in E. coli. Production of secondary metabolites from cultured plant cells. In recent years, it has been shown that a spectrum of compounds can be produced in culture which is beyond the ability of whole plants. By using different precursors, several novel compounds of biomedical importance can be obtained. Pharmaceutical compounds like chiconin is being produced as secondary products with the use of two-stage bioreactor by stimulating the growth phase with the application of different growth regulators. Serpentine can be obtained from Catharanthus and pseudoephedrine from ephedra. Plant cells can also be used to accomplish certain changes in the structure and composition of some industrially important chemicals. This conversion or conversion by means of a biological system is termed as biotransformation. Example, digoxin, cardiovascular drug produced from digitoxin obtained from Digitalis lanata. The biotech industry takes parts in biopharma, industrial biotech, agricultural biotech, food biotech, environment, and bioinformatics. Thank you.